Welcome to our YouTube channel. In this video, we will discuss a question on the serological diagnosis of infections, focusing on the detection of pathogen-specific antibodies in serum. Let us begin with the first part of the question. Infections result in production of pathogen-specific IgM and IgG antibodies in infected individuals. These antibodies can be measured by using different assays. Name the type of tube or bottle that is required to collect a blood sample for studying antibodies in serum. When we need to study antibodies present in serum, blood should be collected into a plain tube, without an anticoagulant. The blood is allowed to clot, and after centrifugation, the separated serum can be used for serological testing. The next question asks us to name 4 assays that can be used to detect antibodies in serum. 4 examples are Enzyme-Linked Immunosorbent Assay, Indirect Immunofluorescence Assay, agglutination tests, immunoblotting, or Western blot. Let us now look at one of these techniques, indirect ELISA. This diagram shows the basic mechanism of an indirect ELISA used for antibody detection. In the first step, the specific antigen is attached to the surface of the well. In the second step, the patient serum is added. If the patient serum contains antibodies against that antigen, these antibodies will bind to the antigen. In the third step, an enzyme-linked secondary antibody is added. This secondary antibody binds to the patient's antibody. Finally, in the fourth step, a substrate is added. The enzyme converts the substrate into a colored product. Therefore, development of color indicates that the patient's antibody has been detected. Here is an example of an ELISA microtiter plate. When interpreting an ELISA plate, we should not simply look at whether a well appears colored or not. We first need to identify the negative controls and positive controls. The negative controls establish the background level of the assay, while the positive controls confirm that the test system is functioning correctly. The patient samples are then compared with the appropriate cutoff or control values to determine whether the result is positive, negative, or, depending on the assay, equivocal. Therefore, proper interpretation of an ELISA requires consideration of the controls, optical density or color intensity, and the manufacturer's cutoff criteria. Now let us consider the clinical scenario. A 9-year-old child was brought with features suggestive of an atypical pneumonia. A Mycoplasma pneumoniae antibody assay was carried out to determine whether Mycoplasma pneumoniae was the cause of the infection. Name the test carried out. The test shown is a particle agglutination assay for Mycoplasma pneumoniae antibodies. In this test, particles coated with Mycoplasma pneumoniae antigen are mixed with serially diluted patient serum. If specific antibodies are present, they react with the antigen-coated particles and produce agglutination. Next, let's look at the particle agglutination assay used to measure the Mycoplasma pneumoniae antibody titer. Across the plate, the patient serum is diluted step by step, from 1 in 2 up to 1 in 2048. In a positive reaction, antibodies bind to the antigen-coated particles, producing visible agglutination, with the particles remaining spread across the bottom of the well. In a negative reaction, agglutination does not occur, and the particles settle into a compact button or dot at the bottom of the well. The antibody titer is the highest dilution of the patient serum that still shows a positive agglutination reaction. For example, if agglutination is present up to a dilution of 1 in 320, but absent at 1 in 640, the antibody titer would be reported as 1 in 320. However, the exact titer considered positive depends on the particular test kit and laboratory protocol.